All right. Um, Hi, I'm Dr. Joanna Wozniak-Brown. I'm the Assistant Director of Resilience Planning at the Connecticut Institute for Resilience and Climate Adaptation. I'm pleased to welcome you to the fourth in a series of webinars about climate change and public health. Today, we'll hear about the connections between vector-borne disease and climate change. Our presenter today is Dr. Shannon LaDeau from the Carey Institute of Ecosystem Studies. Dr. LaDeau is a disease ecologist who investigates how species interactions and environmental conditions influence the spread of pathogens. Current projects include work on how climate change regulates tick populations in Lyme disease, the ecology of mosquito vectors in complex urban riskscapes, and viral transmission in wild fish hatchery networks of the Pacific Northwest. If you have questions throughout the webinar, please post them in the chat box and we'll answer them at the end of the webinar. As a reminder, the presentation is being recorded and you'll be able to view this webinar and all of Circa's webinars on our YouTube channel following the event. So just to let you know that this is the fourth in the series and we have 2 more coming up. So we hope that you can join us for the other ones. And now I'll turn it over to Dr. LaDeau. I'll pass the ball over to you. There we are.
Great, thank you. And hopefully my presentation has now come up for you all.
Looks great.
Great. Well, thank you all for signing in. This is always an exciting time to try to connect virtually. As Joanna said, I'm from the Carey Institute. We're in Millbrook, New York, and my work predominantly is focused on understanding how ecological processes influence disease transmission and risk. In most of my work is human-dominated landscapes. And today, of course, I'm going to talk predominantly about vector-borne disease and climate change. So first of all, what are vector-borne diseases? I want to kind of just make sure we're all on the same page because vector can mean many things. So vector-borne diseases in this talk are caused by pathogens that are transmitted to humans or another animal through the bite of an arthropod. We generally think of this as insects or bugs, but also ticks, mosquitoes, of course, fleas, triatomines, so kissing bugs, these sorts of organisms that require a blood meal of some sort from a host in order to reproduce, generally. Vector-borne diseases have zoonotic origins, which means they all, okay, so meaning that there is a non-human animal at the base of the pathogen system. So in this figure, what I'm showing here is a bird that would be the predominant zoonotic host for a pathogen, and a mosquito, again in this figure, would pick up that pathogen when it bit the bird, and a human would only be exposed if that same mosquito then bit a human. So you require ecological transmission with a non-human animal constantly resupplying the pathogen before you get human transmission. This is how all of our vector-borne diseases that we know about originated. And in this figure here, I just show also that for Lyme disease and tick-borne disease, that's also the case. Ticks are more complicated because they have these very specific different life stages. Here on the right, in the summer, you've got the larval stage, which is the first after the egg stage for the tick, and it generally takes a blood meal from very small animals and can pick up Borrelia, or the pathogen that causes Lyme, if it bites usually a mouse in this part of the country. So in ticks, the blood meals come at each of these life stages, and they require a blood meal in order to move to the next life stage. They can pick up something like Lyme disease, the agent that causes Lyme disease, at any of those life stages if they bite an animal that has that pathogen in its system. Humans don't hold enough of the bacteria that causes Lyme to infect a tick. So adult ticks aren't picking it up from humans. But by the time a tick is an adult, it's already had 2 other blood meals during which it could have got the disease. So it's a much more complicated life cycle and transmission system because there are multiple hosts, but the origin is still a zoonotic, non-human animal where these vectors have to pick up the pathogen that's a disease-causing agent. So some of these vector-borne diseases will continue to require that non-human animal in order to persist. These are the ones where humans do not produce enough of the virus or the bacteria in their own bloodstream to then reinfect another vector. These tend to have less epidemic potential because you're not getting human-to-human transmission, but they're also very difficult to eradicate because you have this reservoir. In this case, if you wanted to eradicate West Nile virus or triple E, you would have to really understand what exactly that zoonotic host is and stop the mosquito from feeding on that host at that stage in order to get rid of one of these zoonotic diseases. And of course, that's very difficult for many reasons. However, several zoonotic pathogens have over human history become directly human transmitted pathogens. These include things like malaria, chikungunya, dengue, yellow fever, Zika. Sometimes they still have non-human reservoirs, but these are all now capable of being transmitted from one human to a vector and then onwards to other humans. This, of course, is what we think of as epidemic potential. These are where you worry increasingly in urban areas that you might get these outbreaks where you can get local transmission that really blossoms over multiple humans. They are, however, in general, easier to, in theory, eradicate locally because you just have to interrupt the transmission between the mosquito and the human. So if you can put humans behind screens, you can have a big effect on reducing yellow fever, which is what we saw after World War II in the US. You could separate humans and mosquitoes more effectively. You could cut down local outbreaks. Notably, all of these arboviral, so viruses transmitted by mosquitoes in the right-hand panel, chikungunya, dengue, yellow fever, and Zika, they're all transmitted by predominantly by one and actually by increasingly two species of mosquitoes. Now, there are thousands of species of mosquitoes, and so it's kind of important to note that only two of them really are responsible for this transmission that is of epidemic potential. They're both Aedes species, and they both increasingly adapted to live very close to humans. In the Northeast, we don't worry about Aedes aegypti as much because it is very sensitive to winter temperatures. It doesn't survive easily from one season to the next above Florida. However, Aedes albopictus has spread globally since the 80s when it left its native range in Asia, most likely aboard tire exports destined for urban areas across the globe. Aedes albopictus in its native range develops through the juvenile stages, which are aquatic dependent, in very small tree holes and in the things like these bamboo cups. The eggs are laid on surfaces that are likely to become wet, and the eggs can persist for months. I've actually had eggs persist for over a year in my lab and still be viable. So they can lay their eggs in things like tires, which then get shipped out and hatch readily in their new environment. And also because of this because they have evolved to develop as juveniles in these very small container habitats, they're also well adapted to small ephemeral container habitats that we see in more urban areas. Aedes albopictus in its native range was not a predominantly human biter. As we see it spread and move into urban areas more globally, it certainly is an aggressive human biter, and it has been responsible for outbreaks of chikungunya and dengue in temperate areas in Tokyo, and in recently, actually, they're assigning some dengue cases from Italy to Aedes albopictus in that region. So it is a competent vector, and it seems to be changing in ways right now that are allowing it to adapt to urban living very close to humans, meaning that it's also likely to continue to become a more important vector of disease in humans. In Connecticut, this is a county map of Connecticut. These data are from Ted Andreadis and others at the Connecticut Experimental Agricultural Station. And what you see here are Aedes albopictus numbers. Those are the circles. The bigger and darker they are, the more Aedes albopictus have been found in that part of the state. What you can see is that it's definitely more abundant in the southern part of the state, and it does seem to be more likely to be found in northern parts of the state in urban areas. So we know that this species does have some temperature sensitivity, whether that's failure to survive through the winter or something, I mean, it's most likely has something to do with the winter temperatures or winter duration, but it's also growing exponentially, and so it takes time for it to continue moving northward. We don't know exactly how far north it will be able to survive, but it has been gradually moving northward. So why is climate an important factor in vector-borne disease risk? I've just given some examples using Aedes albopictus and its northern range limit. Arthropods or ectotherms, all of their bodily processes are determined by climate, by temperature predominantly. So they're very sensitive to temperature. When things get warmer, their rates tend to speed up. So from an internal growth rate at the juvenile stage all the way up through population growth at that scale, those tend to be faster when temperatures are warmer. Now, of course, if I were giving this talk in more southern parts of the hemisphere, we would also be talking about warmer temperatures prohibiting survival because it would be getting too hot. But in the temperate region, in general, warmer temperatures speed up growth and actually favor more population growth for arthropods. So they grow more rapidly, and the population's growing more per season. You get actually often more cohorts per season. For mosquitoes, one thing that becomes important as temperatures get warmer is that several of these species that do well in more human-dominated landscapes can actually grow from egg to adult stages in less than a week. Under the right conditions, with enough resources, Aedes albopictus could grow from egg to adult stages in 4 days. This is potentially problematic when stormwater structures and water management has been designed more around a culex life cycles, which is another genus of mosquito, and with the idea that mosquitoes generally take about a week to develop. So a lot of things have been developed, a lot of stormwater infrastructure has been developed to drain in about a week. People are often taught to make sure all their water habitat in their yards is emptied every week. And especially with albopictus, a week might actually be too long in the heat of the summer. So the risk of human exposure is directly related to the size of the vector population in habitats where people are present. And here's a visual just kind of demonstrate that. If you've got vectors, whether they're picking up a pathogen from a non-animal host, a non-human animal host, or from a human, the more vectors you have, the more likely one is to actually bite that particular infected host. And the more likely they are to then survive and bite another host. So numbers are hugely important. Where you have bigger populations of vectors, you generally have higher risk of disease transmission if an infected host is introduced. For ticks, and we know that populations grow faster as it gets warmer. For ticks, it's a little less clear. First, the research is a little newer in some ways than the mosquito work, which people have been working on for a long time. Ticks and tick-borne disease has really grown both in detection and in funding relatively more recently. And they're also dispersing more. So you get a bunch of species that are also dispersing and moving both north and west, predominantly in this area. And so tying them to temperature becomes difficult because it can be hard to disentangle. For example, the black-legged ticks move northward, whether its dispersal distance is determined by how fast it can move on a host, and that's what's limiting its northward expansion, or if it's actually responding to climate. So there have been some lab studies to look at how changes in temperature influence ticks, and they generally support the idea that ticks are arthropods and they have some thermal limits. If it gets too cold or too hot, they don't function well. A recent effort that I've been involved in with some collaborators, Mary Killaley, Jesse Brunner, Elizabeth McGovern, and Rick Osfeld, that's funded by CERTIP, the Department of Defense funding, has been looking at what happens when you put ticks in their natural habitat and sample them periodically throughout a season. So you can see when during a season they are actually sensitive to temperature or to drying. We put them out, and when I say in their native habitat, we pull up a soil core, we put a bunch of ticks in the soil core, we make sure it's wrapped up with mesh that they can't climb out of, and then you put out 50 and you sample 2 every week. In general, we have these at Fort Drum, so in northern New York, just above the Ixodes range limit, around Millbrook or West Point, and then just south of the predominant Ixodes limit down at Camp Lejeune in South North Carolina. And one of our experiments using these has shown that if you take ticks from the same place and you put them at these different locations, they do survive better from, these are from larval to nymph stages. They do survive better in the warmer, more southern location than in the northern one. So there is a temperature effect in the north that is reducing survival. We also then took similar experimental microcosms into the lab and warmed them by 3 degrees, so projected climate change warming. And while that warming, that's the red in this figure here, the warming didn't have any effect at Camp Lejeune, which is the kind of more left bar, but it did at Fort Drum. So in places where it's colder now, warming 3 degrees increased survival. In places where it's colder, survival was generally reduced in comparison to North Carolina, where temperatures were, ambient temperatures were warmer. So ticks do seem to be sensitive to climate, although a little less directly or immediately than is evident for mosquitoes. So again, we do know that the climate is warming, and we know that warmer temperatures create greater growth rates in arthropods in general, and tend to favor higher winter survival of these animals that have to be dormant for some period of the winter. The longer growing seasons, and we do expect and have seen growing seasons to grow longer. That's the green panel down at the bottom. You could see Connecticut is a little darker green, and that is how many more growing season days we have now versus earlier in the 1900s. And these are from Kunkel, and the figures are from his work, and they're shown at the EPA site. And the right-hand panel in the red shows, again, how much warmer it is now than it was earlier in the century. And Connecticut is warmed, and the growing season is longer. Both of those things support greater population growth of arthropods and faster individual growth, which means greater survival through to adult stages. Okay, so climate change is important. We generally think that warming climate is going to support greater vector populations and greater chances for vector-borne disease. But the northeastern landscape also plays an important role in how we understand or predict what that might look like. So arthropods, both mosquitoes and ticks in this case, actually require vegetation for food and to maintain habitat humidity. They don't do well in paved parking lots. Northeastern cities and suburbs are a mix of high human population density and very green habitat. Our urban areas tend to be pretty green, which is generally a good thing. And I don't want to suggest that it's not. However, having green, natural vegetated habitat in the mix of high human population densities does pose some potential for greater risk of exposure to vector-borne disease for many reasons. This is just reflecting on some work that I've done in Baltimore, Maryland. What you see here are two different blocks from two different neighborhoods where you can see that even though I'm in the middle of a city here, there's a lot of green, there's a lot of trees, there's a lot of vegetation. This is generally a really good thing for heat mediation, for stormwater and water quality and air quality. And in general, vegetation cover is a negative predictor of mosquito density. So this is from work a graduate student, Eliza Little, did in my lab. And in general, larval density, so juvenile mosquito density in container habitats across these types of neighborhoods in Baltimore, Maryland, decreased when you had increasing vegetation cover. However, when you were on a block with a lot of derelict properties, so properties that were abandoned, that weren't managed, you actually saw an increase in density. So you get more mosquitoes with more vegetation when you have less human management of that vegetation. And I think a visual is usually very helpful to kind of unpack that. If you look at, so here are these two blocks here, right? And they both look like they have similar green space. But the one on the left, if you kind of zoom in on the bottom corner, as I've done here, what you see is that the 3 row homes at the bottom there, they don't have roofs, and there's a tree growing out of one of them. And then, you know, one of the big trees is behind another of them. And then when you zoom down under that big tree that's in the yard behind the second row home, what you see is a pile of tires. There were 38 tires in this pile when we sampled it. And so even though the blocks looked very similar, and even though they both had what looked like a very positive feature of having vegetation, the vegetation is associated with very different habitat at the ground level. In ways that's consistent with lower socioeconomic areas that don't have the capacity for managing vegetation across abandoned properties are at higher risk of mosquito exposure because they actually have more mosquito habitat. If the same pile of tires was out in a parking lot and receiving sun all day, then they would not be as productive as they are here under vegetation. So vegetation both plays a moderating effect of keeping the habitat cooler, which is needed, especially in urban areas. It also is important because it falls into the habitat, and juvenile mosquitoes grow predominantly on vegetation detritus. And so again, if we think if those lower income neighborhoods generally have more abandoned properties and more unmanaged or derelict properties, we found across 4 years of our work, higher mosquito abundance. These are female Aedes albopictus per night trapped in lower income neighborhoods versus high income neighborhoods, which is the bottom line. And you can see that there's significantly more. The orange arrow kind of off to the left at around 35 shows the nuisance factor that Maryland was considering for when they were worried about Aedes albopictus, Aedes aegypti and albopictus and Zika several years ago, if they found more than this number in a given trap, that would institute a cause for concern for high risk of local transmission potential. And so you can see that even in our higher income neighborhoods, we hit that. The x-axis is date, and so you get by late July, early August, you hit it in the higher income neighborhoods, but you stay up above it most of the season in the lower income neighborhoods. We've also found that in these same lower income neighborhoods where you have unmanaged vegetation, the mosquitoes are getting bigger. When we collect adult mosquitoes, they're same species. And again, we're only talking a kilometer away, maybe 2 kilometers from the other neighborhoods. They're significantly bigger. And they're significantly bigger predominantly in areas of the neighborhood that have higher, this proportion abandoned is how many derelict or abandoned properties there are in a block, what proportion of that block is abandoned. So if you get more abandoned properties, which is associated with all this unmanaged vegetation, you also get, that's where you're getting the bigger mosquitoes. That's of course important because bigger mosquitoes in some lab studies have been shown to have greater longevity, so they live longer, which means they may have opportunities to bite more, and they can also transmit virus more effectively in some cases. And most of the studies with virus are done on dengue. In these neighborhoods, we've predominantly only detected West Nile virus, and there's less known about how this particular trait might influence West Nile virus transmission. So what can we do to manage risk? The challenges are that we definitely know that the climate is warming and that our urban landscapes are growing and also becoming in many ways beneficially greener, but that these things together are likely to support greater vector populations and greater contact rates. We also know that we're getting that the potential for emergence of a new arbovirus increases, that human interaction and movement, global trade and travel, up until recently anyway, have been growing every year. And West Nile virus was introduced in 1999 to the US and it spread rapidly, and it's still with us. We have native viruses that we know are in zoonotic hosts, like Tripoli, that pop up periodically and likely will continue to. And then we have many pathogens that we might not know about yet, but given the introduction of chikungunya and then Zika in the recent decade, we can definitely expect to see more come. So we know that input of an infected host is a concern and not likely to go away, which means that the fact that our vector populations are growing and likely to continue to grow and be enhanced by changing climate should be cause for concern. So what can we do? We are testing some different control and management options. I wish I could show things that have really worked, but so far we're finding a lot of challenges with managing mosquitoes in particular at urban scales and in urban environments. However, surveillance has got to be key. It's very important that public health agencies know what species are abundant, know what species are present, and know something about when they're most likely to be a nuisance to humans. Arthropods biting humans are a potential risk and should be well documented, especially Aedes albopictus, something that we know is out there and has the potential to transmit viruses we already know about. So surveillance is key. Community engagement seems to be really important. Controlling these mosquito species requires a lot of concerted effort across different land use types and across different property boundaries. Private property's a huge component of that, and when you have vectors that can breed in cups and recycling containers to hundreds of adults emerging, it becomes very important that everyone is engaged and understands how you might work together to manage that risk. And when there are neighborhoods where the capacity does not exist for the community to maintain and manage all of the habitat, usually because there's abandoned, vacant, derelict properties, then it's really important that that someone else is able to step in, because it really does only take it only takes a limited number of container habitats and properties to allow a population like Aedes albopictus to grow and to become problematic, and they need to be controlled early. I've had a lot of wonderful people that have helped with the work that I've done, and I should acknowledge National Science Foundation, the Northeast Integrated Pest Management Center, and CERTA, the Department of Defense, as funders of the work. Thank you for listening. I look forward to being able to answer any questions.
Thank you, Dr. LaDeau. That was great. I think there's a lot of important points in that in your presentation. I think one of the things that you mentioned, just to let folks know, if you have questions too, you can drop them in the chat box and or in the Q&A section, and we'll answer them. So one of the things that was particularly interesting is that we have this challenge of managing urban heat islands with vegetation, and yet that can provide a habitat for these vectors to thrive. So how should planners and city officials start thinking about programs to address issues of blight and property abandonment with vectors in mind?
Yeah, so there's the mosquito concept, which the mosquitoes require an aquatic habitat, and so if there's nowhere for them to develop as juveniles in that aquatic habitat, then so basically managing the vegetation so you don't have garbage or standing water. Garbage is a huge predictor. Litter seems to be ubiquitous with humans, and without really aggressive and persistent management at more agency levels, it seems to collect and stay wherever it lands. So, managing garbage, as kind of unexciting as that is as a research result, does seem to be the most important thing. So whether it's a park that has vegetation that is being well managed, or vacant properties where the vegetation is kind of crawling all over the place, it's the container habitat that persists in those areas that is the real problem for mosquito populations, and especially the mosquito population, the mosquito species that we're worried about as vectors. Now, the other component of it is ticks and the growing tick-borne disease that we're both seeing right now across the Northeast, but also expecting to continue to see increased incidence of. We are starting to sample ticks in Baltimore, even in the city. We know that there are a lot of there's a lot of urban wildlife, and ticks are being moved in and out of a lot of these urban parks. It's a little bit harder to know what the options are for managing that. Right now, the best course of management seems to be to make sure people are aware that if they're outside in anything green and wooded, that there's potential for getting a tick, and people need to be careful of that. Again, garbage. We have one paper led by a postdoc in our group, Ilya Fischoff, who found that in a yard that had some forest in the suburban area here in New York, yards that had forest but also had garbage were more likely to have infected ticks. Probably because yards where garbage is collecting are attracting more mice and supporting greater mouse populations, but we don't know the mechanism of that yet. But so again, it seems that it's the mix of the natural vegetation and the human garbage and litter that's a real problem for supporting vector populations.
I think that's a neat point too about the small mammals, because it's easy to say the deer tick, and so we only think of deers as being one of the hosts, but actually the smaller mammals can be a huge part of that, so mice and chipmunks, even especially in the Northeast, and they love our stone walls.
Yeah.
Yes, so that's a great place for them to be hiding. One of the things that has come up, I've noticed from the Carey Institute research, is actually that the Carey Institute had shared a paper about predicting tick populations based on acorn mast, because then they would predict the mice population, and then it would predict the quantity of tick populations. That might be a little bit of a longer term indicator, but are there some other indicators that health departments should be able to see at the start of a season, to be aware of a season that might be particularly bad versus another?
Yeah, so as with the mosquito-borne stuff, more ticks are predictive of greater risk. There tends to be some positive association between Lyme cases reported and numbers of ticks and high tick densities. But predicting what years there's going to be high tick densities is still very difficult. And the acorn work by my colleague at Carey, Dr. Osfeld, has been fairly consistent in showing a pattern of when there are more resources for small mammals and mice, you get greater small mammal survival. Those populations grow, and then the year after that, you see higher tick populations. And when you have higher tick populations, you have higher infected tick populations. But not all places are oak dominated, and so there isn't necessarily a great correlate of what you would do in your maple hickory forest for that prediction. I've seen some data that support a moderate winter, again, having more of an effect on the mouse survival than on the ticks. And so you have to put it off a year, because if mice survive this winter, you have greater mouse populations the next summer. It's actually the summer after when you would expect the tick populations to show a benefit, because the larval populations of ticks tend to feed on the mice predominantly. And so, but most people are infected by nymphs, the next stage of the tick. So you have to wait a whole year.
Something to look forward to, I guess.
Yeah. So you need a lot of data when you've got these lags. You actually have to have lots of years of data.
Yeah, that's tricky. That actually kind of leads into another question: is are there certain data sets that the state departments of health should be trying to set up and establish, so that we can monitor these changes going forward?
Yeah. So Connecticut is actually one of the states that's got some of the better data sets out there, because you have dedicated, the agricultural station in particular, have dedicated long-term monitoring that happens, and it would be beneficial for more places to have that, where you kind of have enough persistent monitoring of the vector populations that you know what the baseline is for how abundant they normally are, for what time of year they're normally most abundant. And most of these things are so variable from location to location that you really need to have the same location sampled in multiple years, so that you can make some comparisons and if you sample a different location, every year and you're only sampling once, then you don't ever know why one year looks different from another. So it's really important to have some concerted and longer term data collection of the vector populations.
That's great to hear that Connecticut has the good data set. We work hard at it for sure with the ag experiment for that in that instance. Is there certain things that you've seen work for public engagement in communicating the risks, especially in urban populations that might not anticipate this being a problem?
That is really hard. We found it's been very difficult. So people who really want to be out in their own backyard and have land and are used to being out in it tend to be more interested and concerned about things like ticks and mosquitoes. But it doesn't actually necessarily make them more at risk of being infected by them. And so how to get to the communities that need both the information, but also the support for managing the habitats is tricky. We've tried to, we've had a couple of different kind of large scale efforts at community engagement and generally, at least in Baltimore for our work, people weren't all that worried about mosquitoes. It just wasn't really high on their priority list of stuff to worry about, even though we could show them that, the mosquitoes in their neighborhood had West Nile virus. And so we knew it was present. We knew there was actually something there that they were likely to be exposed to. It was hard to get people engaged, partially because they have other stuff they're dealing with, and partially because, again, there's so much property that they have no control over, and that I think people get overwhelmed very, very easily, and are less likely to do something on their own property if they feel like, the next 5 properties next to them are the source of the problem and nothing's going to happen there. So it's hard to engage residents without also making bigger scale changes and showing action at the same time. And that was very different than in the neighborhoods where everyone's got a big backyard and they want to be outside, and the mosquitoes are keeping them away from gardening. Those people were like engaged from the start, and wanted suggestions about what to do. So it's a mixed bag, and it really requires different strategies for different socioeconomic and cultural groups.
That seems to be a running theme in climate change participation, and also developing effective planning strategies across the board. We're pretty close to time, but I have one question specifically about triple E. If you had any information about the spread of triple E in Connecticut.
Yeah, I mean, triple E is it's a difficult system. So triple E is Eastern equine encephalitis. The reservoir is a bird, and there are a few birds that are probably pretty good reservoirs, but the data are really sparse, right? So this is a virus that shows up with devastating consequences in the human population very rarely. I believe the last estimates I saw were about a 60% mortality rate. When diagnosed, we don't know a lot about how many people are exposed but never tested. So 60% mortality rate for people who get sick with it. So it's very scary. We tend to get, 5 to 15 cases in the Northeast a year. Michigan's actually been more of a hotbed recently, and again with on the order of numbers of cases you can generally count on your hand, but are scary. And the mosquitoes that we know we can find occasionally with it are not ones that normally bite humans, right? So they're bird biting mosquitoes. Mosquitoes tend to kind of tend to kind of zone in on one kind of animal, and kind of make, get really good at looking for one kind of animal and staying in that particular habitat. So if you've got mosquitoes that generally feed on birds, it's a pretty rare event that they're going to bite humans too, which is why Tripoli and West Nile even are generally pretty rare in humans. But Tripoli is even more rare than West Nile in that we can generally go out and find West Nile and mosquito pools in reliable and predictable places, but Tripoli is much harder. So unfortunately, that's a lot of that's all not very helpful information. It makes it very, very difficult to predict when and where. The best predictor of Tripoli is when the health departments are doing testing of mosquitoes and they find it in the mosquito pool, and then can make general statements about risk at that time in that specific location. So again, surveillance is so key.
That's a great point, and we'll need to be tracking that as things happen over time and making sure that we're adding those data sets too, and incorporating those into our planning decisions and empowering municipalities to share that information, and also to make sure that we're incorporating that in our urban and rural resilient design, to make sure that we're dealing with these multitude of issues. So we'll stay tuned with your research to see how that's spreading. So we're really excited about that. And thank you very much for your presentation. So if folks want to join us for our upcoming events about recreational water and emergency preparedness, you can sign up at that short link in the right-hand corner of your screen. It is National Preparedness Month in September, so we hope you'll join us for those. So thank you very much, Dr. LaDeau. We really appreciate it, and we hope everybody has a wonderful day.
Thank you. Thanks.